work that we have done in sort of understanding the activation of GPCRs. I'm going to start with some brief introduction so that we can get everyone up to speed with GPCRs and their activation landscapes, and then we'll move over to our methods that we have developed. So I think of GPCRs as cell sensory organs because they recognize what's outside the cell, and they transmit that signal inside the cell by coupling to many cell signaling partners like G proteins and arrestins. So like shown here, so they'll typically bind to a hormone outside the cell and then transduce that signal inside the cell, which ultimately culminates into various responses from the cell, like it could be gene suppression, it could be apoptosis, it could be many other things, cell division. So the cell has to sort of respond to that signal. And these receptors are also involved in our own senses, for example, in sight and taste and smell. So they're very important. So for example, if you had coffee today, you actually inactivated your adenosine receptors so that you can stay awake, although we're missing coffee right now. But also, if you had sweets today, you activated your sweet receptor, which is also a G protein coupled receptor. So in our lab, so my group is actually trying to understand the signaling of GPCRs, starting all the way from sequence and using structure as a sort of a stepping stone to understand how these proteins work. So we have many projects working on different aspects, going from this, from the sequence all the way to how these proteins actually interact with G proteins as well as arrestins. In the past about 20 years, there has been amazing progress in understanding the role of GPCRs in cellular signaling. So many of the pathways inside the cell were independently characterized, but now they have been linked to very specific GPCRs, which can sort of activate different pathways depending on which ligand binds to them. And one of the interesting things about GPCRs is that the signaling is very context dependent. So for example, the same hormone can appear at the outside of the cell, but depending on which receptor is present, it can actually activate totally opposite pathways. An example in context is dopamine receptors, which are activated by this hormone dopamine, which is sort of a reward hormone. And it can actually, if it couples to D1 or D5, then you actually get increase in cyclic AMP inside the cell. But if you have, if they couple to D2 and D4, depending on the cell, then it leads to the lowering of the cyclic AMP. And that has totally opposite effects inside the cell. So these GPCRs are able to do these signaling, and we'll see later on how that takes place. And the other aspect besides G protein signaling is also the arrestin signaling that has been classified for many GPCRs now, where for a normal GPCR, you will have some level of signaling both from G proteins as well as arrestins, but you can also have a ligand that might bias the signaling towards beta arrestins, or you could have a ligand that might bias the signaling through G proteins. And the reason why that's important is to actually understand the pharmacological effects of many drugs. So for example, niacin is a very important molecule that you can actually buy at Whole Foods, in the US at least, and other supermarkets, which actually has an anti-lipolytic effect, which goes through G protein pathways, but it has a major side effect. So many people who take it, they actually get flushing effect, so they stop taking it, and that actually comes from arrestin pathways. And various groups have tried to come up with niacin analogs that sidestep this side effect, but it also loses efficacy in the G protein pathways. And the reason, main reason is we really don't understand the structural basis for how the receptor is biasing the signaling towards one versus the other pathways. And there's a similar example, but in the opposite direction for the D2 agonists. So if we had biased ligands that could really signal through one pathway and not the other, we can really have minimized side effects coming from these receptors. And over the past, I would say, 10 years, there has been amazing progress on the structural side of these proteins, which has culminated in sort of these two major accomplishments. This was the first structure of a GPCR bound to a full G protein out of Kobilka's lab. And this is the first structure of a protein, rhodopsin in this case, bound to arrestin. So these are the two signaling partners, the major ones that we know, that bind to these receptors and activate different pathways inside the cell. And based on this structural information, we have pretty good insight on what the active state looks like, or at least one of the active states, and that is sort of opened up new avenues to understand how these proteins work in general. And this is based on all the structural information and some of our modeling as well as other experiments. This is sort of a biophysical chemist's view of GPCR conformations. So if you just focus on the wild type spectrum of conformations, so these are ranked by energy, so the lower energy is usually thermodynamically more stable. So you have typically a constitutively active receptor. We are not talking about rhodopsin, but other receptors. And they will have an inactive state that will be lowest in energy, and then you'll go higher up, you'll have some constitutively active states, and then some of them might be able to couple to G proteins, and then you might get some states that might be able to couple to G proteins as well as arrestins, and then eventually you might find some states that might couple to beta arrestins. So this type of a cartoon would look very different for different GPCRs, but this is a good way to understand what might happen. So for example, we know from crystal structures, for example, one that Kobilko obtained for the beta-2 receptor with a very strongly binding agonist, and he found that it's still crystallized in the inactive conformation, and that is sort of presented here. And we have worked on mutants of receptors where a specific mutant, even at a single site, could inactivate the receptor or could actually activate the receptor, be more constitutively active. And then before the GS bound structure was obtained, Kobilko also obtained the structure of the beta-2 adrenergic receptor bound to an agonist and a G protein mimic, which is a nanobody. And there, in the presence of the G body, G protein mimic, they were actually able to stabilize the active conformation. So that was the first time we saw an active structure appear. So this type of a cartoon is sort of good for us to understand and sort of think about what are the possible scenarios that might occur in a real situation. And when you think of activation of GPCRs, the time scale is very critical when we're doing modeling. So for example, experimentally we know that the activation occurs on milliseconds to seconds time scales. And most of the MD simulations that we have access to, they barely approach 1 microsecond. And some of them, if you're using D-Shaw's computers, you might be able to get to some milliseconds. And in most of those simulations, we have not seen activation. So for example, if you start from the inactive state, no unbiased MD simulation has been seen to take you to the active state. And so what we have taken is a different approach, which is sort of Monte Carlo inspired, where we try to sample all conformations that are accessible to these receptors. But even that search space is huge, so we have had to sort of follow a focus strategy, and that's what we're going to talk about. So the question that we sort of thought about was, can we predict one or more active conformations of a GPCR starting from the inactive state? And given the fact that the active state conformations are usually high in energy, and when we do homology modeling or other structure prediction methods, you always find the lowest energy structure. So you always miss the active states or the high energy active states. So how do we find these high energy active states if you start from the inactive state? So there are a few general principles that have been used in GPCR. So one is homology modeling and some molecular dynamic simulations. Typically, you can sample some structures even with low sequence identity, and you have to run MD simulations to relax them, which would allow you to sort of sample low energy conformations of different how helices pack in these GPCRs. In our method, that is, I'm going to talk about in a minute, it sort of samples the orientation of these helices in the membrane because it starts with the homology model and then does conformational sampling of helix rotations or orientation inside the membrane, and then we followed up with relaxation by MD simulations. But since same GPCR can have multiple conformations, which are usually characterized through many different interhelical interaction patterns, we want to be able to predict those conformations. And the challenge, as I mentioned earlier, is active states usually have high energy, so how do we actually sample those? So in terms of structure prediction, we are not following the folding process because these are membrane proteins, so it's very hard to follow their folding. We are sort of assembling them in the membrane, sort of in silico. And if you look at the membrane environment, it's a two-dimensional environment, and that provides great periodic boundary conditions to sample the conformations of a GPCR. So, for example, a GPCR present in a membrane, it cannot tilt or rotate beyond a certain angle because of the boundary conditions. And that we used to our advantage, which cannot be used, especially for soluble proteins. There, the conformation space is really huge. But at least it is countable in terms of membrane proteins, where you are limited by the membrane. So we originally developed this Ensemble method, which actually samples the conformations of a receptor. By breaking up the receptor, we do the helix bundle optimization first. By building the receptor in multiple conformations, we do that by following this BiHelix method I'm going to talk about. And the way that works is, if you think about all the conformations that are available to these 7 helices in a membrane, even if you were rotating 1 helix at a time by 30-degree increments, you still have to sample 12 raised to the power 7, which is about 35 million conformations. And for each of them, you have to optimize the side chains to get a very accurate energy. And that's computationally very expensive. So I came up with this BiHelix method, which is you just split up the 7 helix problem into many 2 helix problems, which are nearest neighbor helices. And then you can combine the energies of these later on and get back the energies of those, for example, 35 million conformations. Extending this to helix tilts and sweep angle in the membrane. So the helix orientation in the membrane is defined by a helical axis, which is defined as the least moment of inertia axis. And we sample the orientation of that least moment of inertia axis in the membrane. And it has a helix tilt. It also has helix sweep in the membrane. It also has an internal rotation around the helical axis. So we sample all those angles. And if you sample them explicitly, or if you want to sample them explicitly, you still have to sample about 13 trillion conformations. And for all of them, you have to optimize the side chains. So we sidestep that by using the BiHelix method. And in the BiHelix method shown here, you have, even though you have 7 helices, but only 2 helices are present at any given time. And then as the helices are being sampled, the side chains are also being optimized at the same time. And then that way, we're able to then add up the energies of all the BiHelices. If you want to discuss with me how we do that, we can talk about it after this, later this afternoon in the interest of time. But we add up the BiHelix energies and get back the energy of the all 13 trillion conformations or so. And when we rank them, we can sort of test whether we have worked, our method has worked or not. So this was one of the first tests we did just to get the inactive state. So we put the adenosine receptor helices, but in a wrong template. In this case, the beta-2 template. And the starting structure, which is homology structure, was about 2.1 angstroms. By doing this helix optimization in the membrane, we were able to go to 1.4 angstrom structure. And you can see that actually the crystal structure is green and the sampling, the predicted structure is blue. So we were able to get pretty close. And it was also good enough to actually predict the ligand binding site that was seen in the crystal structures. So we were fairly confident that our energy scoring was working and also the conformation sampling was working. But then we wanted to apply it to find actually active states, because we are still ranking by energy, which actually finds inactive states. So how do we do this conformational sampling? So in terms of, we have to use some other parameter. And the only parameter we use from experiment is this TM3, TM6 distance. So these are the 2 helices that have to separate in the intracellular site so that the G protein can couple from inside the cell. And that distance between helices 3 and 6 changes by about 4 to 5 angstroms upon activation. And that's the only parameter we use in terms of figuring out where the active states are. And it's sort of shown here that helix 6 sort of moves out by about 4 angstroms, and this is what we will be using. So what we do is we sample conformations on a coarse grid in the membrane, and those are shown with these blue circles. And when we find these different conformations, these are then selected based on that TM3, TM6 distance to figure out which conformations might be active-like. So for example, this looks more like an active-like conformation. So we will do a local conformational sampling on that to get a more stable active conformation. So we do it in sort of 2 steps. not advancing. Let's see. Okay. Okay. So we first did the academic exercise, which is if we start with the active state, can we get back the active state? Or if we start with the inactive state, can we get back the inactive state? And we were able to do that, so I'm not going to talk about that. We can discuss that later. So I want to talk about is the real world scenario where you start with the inactive state and see, can we predict the active conformations? And we did that in two ways. One was taking the helix shapes all from the inactive conformations, and the second one was building a hybrid template that builds in the helix shape of TM6, because the TM6 helix shape changes a little bit upon activation, and we wanted to see whether that improves our prediction a little bit more. So, but using both of these methods, we were able to predict active conformations. And this is the sampling of the conformations for the beta-2 adrenergic receptor, where we know the active state, we also know the inactive state. So we started with the inactive state that is shown here, and with the blue circles is the coarse grid conformational sampling, and that was able to identify this low energy conformation in the helix 3-6 distance space, which looked like an active conformation. So we used that as a starting point for the fine grid conformational sampling, and that was able to bring us to conformations that were pretty close to the active conformation, which is about 1.86 angstroms. And using the hybrid template that builds in the helix 6 shape for the active conformation, we're able to go even lower further to about 1.4 angstroms in terms of CRMST. So we're able to actually start from the inactive state and get the active conformations that have been observed experimentally. Then we wanted to see, I asked my student, are there any conformations in this that might bind to the arrestin but not to the G protein? And she was able to find one conformation that can bind to arrestin, which is shown in blue here, but it does not bind to the G alpha because it clashes with it. So we do have conformations in our spectrum of predicted states that can only bind to arrestin. So we are finding more conformations, obviously, that have been observed experimentally, mainly because experiments are very hard to do in terms of identifying other conformations. And in terms of where it appears, it's sort of the arrestin binding conformation is here. So it's not too far in energy. It is accessible. It just has a different conformation. And then using the same helix 3, helix 6 distance, we wanted to see, can MD simulations tell us more about what the activation landscape looks like. So we did 4 to 6 different simulations. So this was the agonist-bound inactive state, or without the agonist, and this was the active conformation, both with and without the G protein and also with and without the agonist. So we wanted to see in different scenarios, what do we actually observe. And we, for each of the scenarios, we actually calculated the energy of the receptor plus the energy of the receptor with the interaction energy it was getting with the ligand, which is the G protein or the agonist. So this actually exclamation is actually a minus. So sorry, it didn't reproduce. So if you look at the average energies of these states, what we find is that, so the red one is the APO conformation, and when you look at the energy landscape, it has some energy, but when you have the agonist that's bound to it, it actually lowers the energy of all of them. Yes, okay. All right. So the main question, conclusion here was that the agonist bound state is still like the inactive state, but when you have a G protein present, then the active conformation is favored. And we applied this method then to another receptor, somatostatin receptor 5, and you got very similar results. So you have these antagonist binding conformations and agonist binding conformations on this side. And there also we got a very similar landscape. So you got the active conformations that were only stabilized by the G protein. And this was just to show that if you don't include the interaction energy with the G protein, then the receptor by itself is actually highly strained in the active conformation. So this is just to show that our conformation was stable of the somatostatin receptor bound to the G-alpha I. And then we actually looked at the helix 3-6 distance. When you have the apo, there's nothing that happens to the 3-6 distance, but when you have the agonist bound, without the G protein, actually you form a 3-6 salt bridge, and that will definitely do not allow it to couple to the G protein, but with the G protein bound, obviously, they both behave the same way. So I'll skip this. So we have a method called active Ensemble that allows us to predict multiple GPCR conformations, and many of them are active, and they actually can have different binding profiles to different signaling partners. And it also provides, the method enables new insight into how these proteins activate. So this grad student did most of the work, and Adam and Vaclav, they helped with some of the scripts. And I'll leave you with this message, and if you have more questions on my poster, you can visit me this evening. Thank you.
We have time for a question.
Hello, thank you very much for the presentation.
Yes.
The question is, what force field did you use during the MD relaxation stage?
So the MD was done with the charm force field. Charm.
Okay, thank you very much.
Sorry. You did simple MT, you didn't try a replicate change method, for example, for sampling?
No, we did simple MD.
Okay, I think we better move on. So thank you very much. Let's thank our speaker again.